Okay. First question. In dash type of inflorescence, the main axis terminates in a flower, hence is limited in growth, and flowers are born in dash succession. Okay, if the main axis terminates in a flower, if it is showing unlimited growth, it is a racemose type. If it is terminating in a flower, it is a cymose type of inflorescence. So in cymose type of inflorescence, main axis terminates in a flower, hence is limited in growth, and flowers are born in a basipetal succession. That is, old flowers at the young flowers at the base and old flowers at the tip. That is called basipetal succession. Okay, but in case of racemose type, okay, we'll find the main axis continues to grow, and we'll find acropetal succession. That is old flowers at the base and young flowers at the tip. Okay. And next, second question. Match column 1 with column 2 and select the correct option from the codes given below. Column 1, column 2. Okay, column 1, pedestal. Pedestal means the stalk of the flower. Okay, the stalk of the flower is called a pedestal. And next, peduncle. Inflorescence axis. Okay, the axis of the inflorescence we call it as peduncle. Okay, bract. Bract is nothing but a reduced leaf. Okay, bract is nothing but a reduced leaf. Whereas a petiole is a stalk of the leaf. Petiole is a stalk of the leaf. So we see. So that is a option. And next, the third question. In dash flowers, the margin of thalamus grows upward, enclosing the ovary completely and getting fused with it. That is the epigynous flowers. Epigynous flowers or inferior ovary. We can also call it as inferior ovary, because the ovary is present below, and all other floral parts are arising away above from the ovary. That's why we call it as epigynous flower. So in epigynous flowers, margin of thalamus grows upward, enclosing the ovary completely and getting fused with it. Okay, so that is epigynous flower. And next, find out the incorrect match. Okay, sterile stamen, we call it as a staminode. Stamens attached to petals, epipetalous. Stamens attached to perianth, epiphyllous. Okay, the term that is used, if stamens are attached with the perianth, we call it as epiphyllous, not episepalous. Okay, because perianth means okay, the sepals and petals are undifferentiated, so we don't have sepals and petals. That's why we can't use this term sepalous. So we use the term epiphyllous. And next, free stamens. Okay, free stamens, polyandrous. Okay, free stamens are not called as a polyandrous. Polyandrous means if stamens are present in groups, in many groups of stamens are present, then we are calling it as or many bunches of stamens are present, then we are calling it as polyandrous. Sorry, polyandrous means a free stamen. Okay, free stamens we call it as a polyandrous. Okay, if they are fused, we call it as adelphous, monoadelphous, diadelphous, like that. So the incorrect statement is C option. Stamens attached to perianth, actually we have to call it as epiphyllous, but it is given as episepalous. And next, fifth question. If the gynoecium is present in the top most position of the thalamus, the flower is referred to as Hypogonus, because as the ovary is present at the top and the remaining floral parts are arising below the ovary, we are calling it as a hypogonous flower. Okay. And next, ovary is one chambered but becomes a two chambered due to the formation of a false septum in brassica. Okay, such a placentation. Okay, such, okay, we observe this placentation. Okay, this in case of free central. In case of free central type of placentation, we'll observe, okay, this arrangement. Okay, it is observed in a brassica. And next, in placentation, in dash placentation, a monocarpellary ovary bears a single longitudinal ovule along the junction of the fused margins. Okay, that is a marginal placentation. Okay. Okay, that is okay. We'll find that two margins were fused on ventral side. At that region, we'll observe the arrangement of ovules or seeds, and such a type of placentation we are calling it as marginal placentation. And next, select the incorrect pair out of the following. Okay, monoadelphus hibiscus. In hibiscus, we'll find all the stamens are fused and they are present in one bunch. Okay, and next, diadelphus cucurbita. In okay, it is a polyadelphus in cucurbita, not diadelphus. The diadelphus is observed in a, okay, diadelphus is observed in a, okay, beans, peas, grams, like that. Okay, in Fabaceae. And polyadelphus citrus X. Syngenesious, that is a Helianthus anus. Syngenesious means okay, the anthers are fused, not the filaments. The anthers are fused. That's why we call it as syngenesious. It is a sunflower. Sunflower. In sunflower, we can observe that. Helianthus. The Helianthus anus is the scientific name of sunflower. And next, in dash estivation, sepals or petals. In estivation, in dash estivation, okay, sepals or petals in a whirl just touch one another at the margins without overlapping, as it is found in a, okay, that means the type of estivation is valvate type, and a valvate type of estivation is observed in a, okay, is observed in a calotropis. Type of estivation is observed in a calotropis example. And next, ovary is said to be half inferior in which of the following conditions? That is a perigynous. In perigynous flowers, okay, the ovary is said to be half inferior or half superior. We can also call it as, okay, in a perigynous flowers, we can observe such phenomena. And next, read the given statements. Gynoecium occupies the highest position while the other floral parts are situated below it. Okay, gynoecium is present at a highest position and the floral parts are situated below it, means it is a type of hypogynous flower. Ovary is superior, yes. Okay, ovary is superior in hypogynous flowers. Examples are brassica, hibiscus, brinjal, etc. So examples for hypogynous flowers. Okay, which condition of the flowers being described by the above statement? So the above statements are related to hypogyny, that is hypogynous. And next, match column 1 with column 2 and select the correct option from the codes given below. Column 1, column 2. Okay, marginal presentation. Okay, example for marginal presentation. P. In peas, grams. Okay, we'll find this marginal presentation. And next, parietal presentation. Okay, parietal presentation. Okay, we can observe in case of okay, mustard and argemone. Okay, mustard and argemone, that is prickly poppy. Okay, and next, axial presentation is observed in case of hibiscus, tomato, lemon. Hibiscus, tomato, and lemon. And next, free central type. Okay, the free central type is observed in dianthus. Okay, in dianthus and primrose will find free central. And basal type of presentation is observed in sunflower and marigold, or sunflower, marigold. In all this, okay, and grasses will find this basal type of presentation. Okay, so the option is A. Correct option is A. And next, 13th question. Okay, syngenesious condition of stamens is found in family Asteraceae. That is, we said no. Okay, sunflower. Okay, in case of sunflower. Okay, and next, in case of Tridax. Okay, Tridax. Okay, Asteraceae. Okay, in Asteraceae we'll find okay gold, sunflower. Okay, and next chrysanthemum. Okay, Tridax. These are all belongs to Asteraceae family. In this Asteraceae, we can observe this syngenesious condition. That is, the anthers are fused. And next, flower is complete when it has calyx, corolla, androecium, and gynoecium. When all the floral parts are observed in a flower, then we are calling it as a complete flower. And next, a plant with both male and female flowers is monoecious. Plant. Okay, that means here the flowers are unisexual, but both male and female flowers are born on the same plant. Okay, in that condition, we called the plant as bisexual, but the flowers are unisexual. In that condition, we use this term monoecious. And next, choose the correct option for A and B. Okay, here one is A is a racemose type of inflorescence, and B is a cymose type of inflorescence. A racemose and B cymose. Option 2. And next, which of the option is correctly matched with the diagrams? Okay, A is valvate type, B is a twisted type, and C is a imbricate type, and D is a vexillary type of estivation. So valvate, twisted, imbricate, and vexillary. That is option 1. And next, choose the correct combinations. Okay, first one, Hypogonus, Perigynous, and Epigynous. So here, okay, A is a Hypogonus, B is a Perigynous, and C is a Epigynous. So A1, B2, C3. So option A, that is first. And next, what kind of presentation is represented by the given figure? Okay, here the type of presentation is a axial type of presentation. Okay, here we can see the ovary is divided into chambers, and next the placenta is present at the center and which is swollen, and from this swollen placenta which is present at the center, the ovules are arranged. Such a type of presentation we are calling it as axial type of presentation. Okay, it is observed in case of lemon, tomato. Okay, in all that. Okay, that is option B. And next, in an inflorescence where flowers are born laterally in an acropetal succession, the position of the youngest flower bud shall be. Okay, if it is an acropetal succession, it represents okay, racemose type of inflorescence. In racemose type of inflorescence, okay, the young flowers are present at the tip, that is at the distal. Okay, not at the proximal. Proximal means lower. Okay, distal means are at the last. So the young are present at the distal region. Okay. And next, what type of presentation is seen in sweet pea? Marginal presentation. Sweet pea. Okay, sweet pea belongs to Fabaceae. In Fabaceae, we'll find marginal type of presentation. Okay, and next, long filamentous threads. Okay, protruding at the end of the young corn of maize represent styles. Okay, they represents the styles. This is very, very important, ma. Okay, in +2 also we'll come across this one, the styles. Okay, the styles only represent cob of a main. Okay, that is the long thread-like structures that are emerging out of the corn cob. Okay, that the thread-like structures represents the styles because it is pollinated by wind. To capture the wind-borne pollen grains, the styles are long and they are freely placed in the air so that they can capture the pollen grains. And next, epipetalous stamens are found in epipetalous. Okay, we'll observe in case of Solanaceae. In Solanaceae, okay, the filaments of the stamens are attached with the petals of a flower. That's why we call it as epipetalous. Okay, in case of Solanaceae, datura, in case of Solanum tuberosum, Solanum melanogena, Solanum nigrum. Okay, we'll observe this epipetalous condition. And next, an example of axial presentation, lemon. Okay, in lemon we'll find the axial type of presentation. And next, pentamerous actinomorphic flowers, bicarpellate ovary with oblique septa and fruit a capsule or berry are characteristic feature of Solanaceae. Okay, these are the description of a flower in case of Solanaceae. We'll find 5 sepals, 5 petals, 5 stamens. Okay, and next the flower can be cut into 2 equal halves and we'll find 2 carpels which are fused, and the septa is oblique. It is not exactly straight. It will be in an oblique position. Okay, and fruit. Okay, and the type of the fruit we call it as either capsule or berry. Okay, these are all the characteristic features of Solanaceae. And next, assertion reason type questions. Assertion: the flower is a modified shoot meant for a sexual reproduction. Yes, the flower is a modified shoot meant for sexual reproduction. Okay, the flower is described as a modified shoot because we said very clearly. Okay, the flower is a reduced branch which consists of nodes and internodes which are very much condensed. That's why we say that the flower is a modified shoot. Okay, meant for sexual reproduction. And next, flowers are arranged in different types of inflorescence. Okay, flowers are arranged in different types of inflorescence. There we will find racemose type, okay, cymose type, and other types of inflorescence. Okay, that is. But okay, assertion and reason both are correct statements, but R is not explaining the yes. Okay, reason is not explaining the assertion. Okay, that is so B option. And next 27th assertion: actinomorphic flowers show radial symmetry. Okay, zygomorphic flowers are bilateral symmetry. Yes, the statements both A statement and R statement are correct, but R is not the correct explanation for or reason is not the correct explanation for assertion. Okay, and next 28 assertion: petals are usually colored. Reason: okay, petals attract insect for pollination. Yes, Okay, why? What is the major function of petals means? Why the petals are brightly colored means the petals are brightly colored to attract insects for pollination. Yes. Okay, assertion is correct, reason is correct, and reason is the correct explanation for assertion. Okay, and next, assertion. Calyx and corolla are accessory organs of the flower. Reason, they do not directly involve in sexual reproduction. Yes. Okay, assertion is correct, reason is correct, and reason is the correct explanation of a assertion, because as they are not involved in sexual reproduction directly, they were called as the accessory organs, not the essential organs. Okay, and next question. Assertion. In China rose, stamens are said to be monoadelphous. China rose means hibiscus. In hibiscus, we'll find monoadelphous. Okay, the flowers are okay. That means in this the stamens are fused to form as a single unit. All the okay as a single unit. That's why we call it as a monoadelphous. And in China rose, a fusion of filaments of the stamens form a single group. Yes, that's still okay. This means okay, as it is present as a single group, we are calling it as a monoadelphous. So assertion is correct, reason is correct, and the reason is the correct explanation of assertion. Okay, that is. Anybody is having any doubt?
No, sir.
Okay. And next, coming to our topic. Okay. Yesterday we are discussing about ribosomes and inclusion bodies. Okay, we are discussing about ribosomes. Okay, when already we said okay, the units for ribosomes is given as S. Yes, what is this S represents means? It represents a Swedberg unit. Okay, it represents a Swedberg unit. What is this Swedberg unit means? This Swedberg unit represents the okay centrifugation coefficient or sedimentation coefficient. Okay, centrifugation coefficient, or we can also call it as a sedimentation coefficient. That means the units for a centrifugation. Okay, that means okay, you all know the centrifugation process. That means the centrifugation is the process which is used to separate the molecules based upon their density. Okay, that is the process which is used to separate the molecules based on their density. Okay, based upon the supernatant that we are using, or based upon the fluid that we are using, we can separate the molecules which are slightly differed in their density based upon the solution in which we are separating the molecules, and we okay, and the type of centrifugation process, ultra centrifugation, ultra centrifugation, like this we have got. Okay, the revolutions, the number of revolutions that we'll make use of. Okay, that one will depend will helps in separating these molecules. Even a molecules which are slightly variated, very slightly variated in the density, also we can separate by using centrifugation. Okay, and that okay, the units for this centrifugation we call it as Swedberg unit. So when the subunits are present separately, okay, they take okay, that means in a separate okay, in a set okay, set a certain period of time. Okay, it will okay, that means okay, 50 years. Okay, 50 molecules are separated, 30 molecules are separated. So that's why we are calling it as a 50 years and 30 years. But when these two molecules are combined, okay, it is coming at a 70 years instead of 80 years. Okay, the difference is why this difference occurs means because of the reduction in the surface area when these two molecules are reduced. That means that the contact between these two molecules, that surface area is lost and overall surface area will only comes. That's why the centrifugation also a little bit lowered. So it comes to 70 s. Okay, that is. So we'll find, okay, overall in case of prokaryotes, what is the type of ribosomes? Means 70s type of ribosomes. And these 70s ribosomes, okay, 70s type of ribosomes, when they are, okay, they are also having two subunits, that is the larger subunit as 50s and a smaller subunit as 30s. Okay, and next, okay, when you observe the ribosomes, what is the function of this ribosomes? Ribosomes are the sites of protein synthesis, or they are also called protein factories, because ribosomes are involved in protein synthesis or formation of polypeptide chain. Okay, they form the polypeptide chain, which is we also call it as a protein molecule. Okay, that is, so the ribosomes are the site of protein synthesis or a polypeptide chain formation. And next, when you observe here, ribosomes may attach to a single mRNA. Okay, mRNA. When you observe the RNA, there are different types of mRNA. In that, there is a mRNA, that is a messenger RNA. mRNA means messenger RNA. Okay, this RNA is produced from the DNA by a process called transcription. Okay, the process of synthesis of RNA from DNA, we are calling it as a transcription. During this process, what happens means, whatever the genetic information that is present on the DNA, that is copied onto the RNA. So whatever the information that is copied on the mRNA, based on that, the sequence of amino acids that are present in a polypeptide chain or protein molecule will be determined. So to form that polypeptide chain, okay, the ribosome has to attach with the RNA, and this mRNA will read the sequence of that mRNA, and according to that, the new polypeptide chain is synthesized. So in that, okay, in case of prokaryotes, here we are discussing about prokaryotes. In prokaryotes, okay, a single a single mRNA can bind with many ribosomes and form a chain called polyribosome or polysome. Okay, such a mRNA which is associated with many ribosomes. Okay, such a structure we are calling it as a polyribosome, or simply we call it as a polysome. Okay, that is. And next, the ribosomes of a polysome translate the mRNA into proteins. So the ribosomes, okay, that means by using the information that is present on mRNA, according to that, the protein synthesis will occur. Okay, the protein synthesis will occur. Okay, that is okay. That is about the ribosomes that are present in prokaryotes. And next, along with that, we'll also observe certain structures called inclusion bodies. Inclusion bodies. So what these inclusion bodies? Okay, these are also not membrane structures. Okay, they are present as a reserve material. Okay, the reserve food materials. The reserve food materials in the prokaryotic cells will form the inclusion bodies. The reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. Okay, in the form of inclusion bodies. Okay, they are stored, the reserve food material, and these are not bound by a membrane system and lie free in the cytoplasm. Okay, these inclusion bodies do not have any membrane surrounded, but they freely suspend in the cytoplasm of the cell. Okay, that is, we'll find different types of reserve materials, okay, in the form of phosphate granules. Okay, the phosphate is stored in the form of phosphate granules, and cyanophycean granules. Okay, that is the pigment granules. And next, the glycogen granules, that is the carbohydrates are stored in the form of glycogen. Okay, that is. And next, along with this reserve material, we'll find a certain gas vacuoles. That is, different gases are released, different as a result of different metabolic activities. Okay, those are enclosed in vacuoles, and they are called as gas vacuoles. Gas vacuoles are found in blue, green, purple, and a green photosynthetic bacteria. So different types of gases in in a green, in green and purple bacteria, we'll find the hydrogen sulfide gas is releasing. Okay, and in green photosynthetic bacteria, we can observe the release of oxygen. So that is, so these are all enclosed inside the vacuoles, and these are called gas vacuoles, and which will later reaches to the surface, and finally they are released outside. Okay, this gas vacuoles. Okay, that is, these are the some of the inclusion bodies that we observe in case of prokaryotes. Okay, anybody is having any doubt regarding ribosomes and inclusion bodies? And next, coming to the eukaryotic cells. Eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. Okay, that means up to now we have discussed about the prokaryotic cells. Now we'll go with the eukaryotic cells. Okay, which organisms will possess the eukaryotic cells means protistans, and next fungi, plants, and animals. All these organisms possess eukaryotic cells. And in eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Okay, when you observe, in case of prokaryotes, okay, the cytoplasm is not having any compartment. That means we don't find any cell organelles or any membrane-bound cell organelles. We don't find in prokaryotes. But when you come to eukaryotes, in eukaryotes, okay, the cytoplasm is divided into many compartments. Why? Because due to the presence of membrane-bound cell organelles. Okay, due to the presence of membrane-bound cell organelles, okay, we say that the cytoplasm is divided into many compartments. Okay, that is okay. And next, eukaryotic cells possess an organized nucleus with a nuclear envelope. Okay, that means when you observe in case of prokaryotes, the genetic material is scattered in the cytoplasm. There is no particular region. Okay, particular region in the cell where this genetic material is present. Okay, it is scattered in the cytoplasm. But when you observe in case of eukaryotes, in eukaryotes, the genetic material is placed in a certain position, okay, which is enclosed by a nuclear envelope, and which we are calling it as a nucleus. Okay, that is. And next, in addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Okay, and next we'll find some generally when you observe in case of prokaryotes, the locomotory structures are flagella with simple organization only. But when it comes to eukaryotes, we'll find different types of locomotory structures with a complex organization. And at the same time, we'll find some cytoskeletal structures. That means structures that are present on the inner surface of the plasma membrane, okay, which will provide some shape and structure to the cell. These are called cytoskeletal structures, like microtubules, microfilaments, like that. Okay, that is so. So we'll find some complex locomotory and cytoskeletal structures. And next, their genetic material is organized into chromosomes. Okay, their genetic material is organized into chromosomes. Okay, that means usually we'll find more than one chromosome in case of eukaryotes. But in case of prokaryotes, all the genetic material that is present, okay, which is a circular DNA equal to one chromosome only. But in case of eukaryotes, usually we'll find more than one chromosome. That's why we are calling as chromosomes. Their genetic material is organized into chromosomes. In different organisms, different number of chromosomes. Okay, the number of chromosomes will be varying from species to species. Okay, that is. And next, all eukaryotic cells are not identical. Okay, they're not similar. All the eukaryotic cells are not similar. Okay, we'll find certain differences. Okay, for example, you can see the plant and animal cells are different as the former possess cell walls. Okay, former means the plant cells. Plant cells possess cell wall, they possess plastids, and they possess a large central vacuole which are absent in animal cells. So whatever the structures we are saying, that is cell wall is absent in animal cells. Plastids means like chloroplast, which is involved in photosynthesis. Okay, and some chromoplast, which will give colors. Okay, and some leucoplasts, which are involved in the storage of food material. All this we call as plastids commonly, and all these are absent in animal cells. They are present only in plant cells. Okay, and we'll find a large central vacuole, which is the characteristic feature of plant cells. So all these are the cell wall, plastid, and a large central vacuole are absent in animal cells but present in plant cells. Okay, but okay, and next when you observe here, on the other hand, animal cells have centrioles which are absent in almost all plant cells. Okay, when you observe here, okay, the centrioles, okay, the centrioles which are involved in the formation, okay, in the formation of spindle fibers or spindle apparatus, and involved in cell division, and these centrioles are usually absent in plant cells but present in animal cells. Okay, okay, but absent in plant cells. Okay, the centrioles which are involved in cell division. Okay, you can see here. We can see the plant cell and animal cells. Okay, this is the plant cell. You can see, okay, the cell wall. Okay, cell wall, and we'll find the plasma membrane. And next, inner to the plasma membrane, we'll find the cytoplasm. This is all cytoplasm, and this is the nucleus, which is surrounded by nuclear envelope or nuclear membrane. Okay, and the centrally concentrated dark color region we call it as a nucleolus, and this whole region we are calling it as a nucleus. Okay, and when you observe here, the cytoplasm, we said very clearly, because the plant cells are eukaryotic cells, which is divided into many compartments. We can see here the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi complex, okay, mitochondria, chloroplast, okay, ribosomes, okay, and next peroxisomes, okay, and we'll find a large central vacuole. Okay, because of the large central vacuole, the nucleus is pushed towards the peripheral region, towards the corner. Okay, generally in case of animal cells, the nucleus is present at the center of the cell because they do not have a this large central vacuole. That's why usually the nucleus is present at the center. But in case of plant cells, the nucleus is pushed to a side, to a corner. Okay, that is not present at the center because of the presence of this large central vacuole. Okay. And next, okay, that is okay. When you observe the plant cells, okay, this is the cell wall of this plant cell, and this plants, okay, cell wall of the adjacent cell. That means the other cell which is present other on the opposite other side. Okay, in between we'll find a layer called middle lamella. This middle lamella will act as a cement-like substance and joins the adjacent plant cells. So we'll find a middle lamella. Okay, towards the inner side, we'll find the cell wall, and towards the inner side of the cell wall, we'll find the plasma membrane. So the middle lamella is the common layer present between the adjacent cells. Okay, that's what we can see here. So middle lamella, which is present at the center, and this is the cell wall of this plant cell, and this is the cell wall of the adjacent cell. So in between we'll find the middle lamella. Okay, this middle lamella will act as a cement-like substance, and we'll find the chloroplast, peroxisomes, and this is the cytoplasm. We'll find ribosomes and a large central vacuole, which is the characteristic feature of plant cell. And then the nucleus is surrounded by a membrane called the nuclear envelope. And we'll find the nucleus and the nucleolus, the dark stained, okay, centrally located region of the nucleus, we call it as a nucleolus. Okay. And we'll find a rough endoplasmic reticulum, smooth endoplasmic reticulum. That is, a rough endoplasmic reticulum means that the reticulum on which, okay, on which we'll find ribosomes on its surface. We are calling it as rough endoplasmic reticulum, and we'll find lysosomes. Okay, Golgi apparatus we can observe. Okay, that is. Okay, we'll discuss all these structures. And but when it comes to plant cells, okay, but when it comes to plant cells, in plant cells we don't the large central vacuole, we don't find a cell wall, we don't find a large central vacuole, we don't find a plastids or a chloroplast. Okay, but we'll find the nucleus usually present at the center, which is surrounded by nuclear membrane, nucleus, and the next we'll find the nucleolus, nucleus surrounded by nuclear envelope or nuclear membrane. And here also the cytoplasm is present. Okay, thick and fluid like a cytoplasm, which is rich in nutrients. Okay, which is divided into compartments due to the presence of membrane bound cell organelles. Okay, we can observe rough endoplasmic reticulum, smooth endoplasmic reticulum, and next we'll find a Golgi apparatus or Golgi complex. Okay, and next we'll find mitochondria, but the plastids are absent. We'll find peroxisomes, lysosomes. Okay, lysosome, peroxisome. We'll find ribosomes. Okay, ribosomes are freely present in the cytoplasm, some, and some are associated with the endoplasmic reticulum as a rough endoplasmic reticulum. Okay, and in plants, okay, in animal cells, we'll find the outer, the delimiting structure is the plasma membrane. We don't find cell walls. And what are the structures that are present means centrioles. We can see these centrioles are present usually near the nucleus. These centrioles are mainly involved in a cell division and forming a spindle apparatus, by the formation of a spindle apparatus. Okay, that is. And we already said there are, okay, in animal cells, some of the free surface, okay, some of the animal cells on its free surface will find small projections towards the outer side, and these are called microvilli. We can see microvilli here. Okay, these are the some of the differences that we can observe with reference to plant cell and animal cell. Okay, coming to the detailed structure of the eukaryotic cell. Okay, let us now look at the individual cell organelles to understand their structure and functions. Okay, the different cell structures that are associated. The first one is the cell membrane. Okay, cell membrane. The detailed structure of membrane, that is the cell membrane, was studied only after the advent of the electron microscope in the 1950s. That means, okay, that means the detailed structure of the cell membrane or plasma membrane was studied only with the discovery of a electron microscope. Okay, with the discovery or with the invention of a electron microscope, okay, in the year 1950, from okay, then only we came to understand the detailed structure of plasma membrane. Okay, meanwhile, okay, that means before this, what they have done means, meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. So that means that means before 1950s, okay, before the discovery of electron microscope, we don't know, okay, the detailed structure of plasma membrane, but we came to understand the chemical composition of the plasma membrane. Okay, chemical composition we can understand now. So the chemical composition of the plasma membrane was studied before the detailed structure of the plasma membrane was studied by using electron microscope. So because of the already, as we know, the chemical composition of the plasma membrane, it was made easy to understand the detailed structure of the plasma membrane. Okay, by using certain cells, especially human red blood cells. Okay, human red blood cells. Okay, human red blood cells also consist of plasma membrane now. So they are okay, that means this human red blood cells are very easy to extract, and we can easily understand the chemical nature of the plasma membrane. So that is so detailed structure of the plasma membrane is known. Okay, we have known the chemical composition of the plasma membrane, and later we have described the detailed structure of a plasma membrane in the year 1950 because of the discovery of a electron microscope. And next, these studies showed that cell membrane is mainly composed of lipids and proteins. So that means, okay, with the with the invention of a electron microscope, we came to understand the detailed structure of plasma membrane. Okay, when they are when they are studying about the structure of a plasma membrane, what they came to understand, that means, okay, the plasma membrane is mainly made up of lipids and proteins. So the major lipids are phospholipids. That means the type of the lipids that are present in the major types of lipids that are present in the plasma membrane are phospholipids, conjugated lipids, the lipids that are associated with certain compounds like a phosphate. That's why we are calling it as a phospholipid. So the major lipids are phospholipid. That means that are arranged in a bilayer. That means these phospholipids are arranged in a two layers, outer layer and inner layer. Okay, that is. And next, when you observe here, when you observe this phospholipid, okay, as the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner part. Okay, when you observe here, we can observe very clearly. Okay, this blue color structure and this black color thread like structures are there. Okay, these, okay, all this structure together, this blue color round ball like structure and this black color thread like structures, okay, this all together we are calling it as a phospholipid. Okay, it is having a, okay, hydrophilic head and hydrophobic tail. Hydrophilic head and hydrophobic tail. Hydrophilic means which can accept water. Okay, which can accept water. And next, hydrophobic means which cannot react with water. Hydrophobic, that means they are anti, opposite. That means they do not dissolve in water. That's why we say it as a hydrophobic. Okay, the tails. Okay, in a phospholipid, we'll find two structures: head and a tail. The head is a hydrophilic, whereas the tail is a hydrophobic. Understand? Okay, when you observe here, so this is the head, which is a hydrophilic, and these are the tails. Okay, hydrophilic tails. Hydrophobic tails, which are present towards the inner side, whereas the hydrophilic heads are present towards the outer region on both. Okay, that is this is one layer. Okay, this is one phospholipid layer, and this is another phospholipid layer. That's why we are saying the plasma membrane is consisting of a lipid bilayer. That means the lipid molecules are arranged in two layers. One layer towards the environment and one layer towards the cytoplasm of the cell. Okay, and both the ends, what happens? The hydrophytes are present towards the outer region, whereas the hydrophobic tails of both the phospholipids are present towards the inner side. Why this hydrophilic heads are placed towards the outer region or towards the environment and towards the cytoplasm means because it facilitates to dissolve in the water and helps in the transportation of substances, and it will not as a result the structure of the plasma membrane cannot be distracted. Okay, if the hydrophobic tails are present towards the outer region, okay, as they are hydrophobic, they can be easily destroyed by this water content of cytoplasm and the environment, because most of the organisms lives in water. So if they are in contact with if the hydrophobic tails are in contact with water, the structure of the phospholipid may get destroyed. So that's why, to prevent the destruction of this phospholipid, the structure of phospholipid, what happens? The hydrophilic heads are present towards the outer region, which can interact with the aquatic environment. Okay, and the hydrophobic tails are present towards the inner side. Understand? Okay, that is what we can see here. Okay, so we see here. So the major lipids that are arranged in a bilayer, that means in two layers. Also, the lipids are arranged within the membrane, and with the polar heads towards the outer sides and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. So that means as the organisms are in contact with the body fluids, or they are in with the outer environment with the water, and the body fluids are also consisting of measure of water, and the cytoplasm also consists of water. So if hydrophobic tails are present towards the outer region, they may get damaged. They may interact with the water, and they may get affected. So to prevent or to preserve the structure of this lipid, phospholipid, usually the hydrophobic tails are present towards the inner side, and the hydrophilic heads are present towards the outer region. Okay, and next, in addition to phospholipids, membrane also contains cholesterols. Okay, small quantities of cholesterol in between. Okay, in the plasma membrane, which are also a type of lipids. Okay, so that is. And next,